Jessica Elisa Garcia from Instituto de Ecología from the National University. So thank you.
Do I need the mic? Or I can speak without it? What do you think? It's okay? Or it's better?
That's up to you.
Wow. Yes. In case you don't hear it well, you can tell me. So first I want to thank the organizing committee for inviting me to this meeting, and particularly to Dr. Javier Juarez, and it's a pleasure to be here. And today I'm going to talk about the two MACBOX genes, Chantal1 and 2, and their implication in flowering transition and meristem maintenance. So MACBOX genes are transcription factors that are characterized by their MAC domain that binds to consensus and non-consensus charge boxes, and to do it they have different configurations, like dimers, tetramers, or in complex with other proteins that makes the DNA can loop and we can cluster the regulatory regions. They are in all eukaryotes, and for example, they are, I'm sure that you have heard about the serum response factor in animals and the myosin enhancer factor of MEF2, that they participate in proliferation differentiation decisions in different cell types, and when they are mutated or disturbed in any way, they produce diseases and cancers. In plants, there are many MAC boxes, around 100 in Arabidopsis thaliana, that is our plant model, and they participate along the plant's life cycle from the vegetative growth to the seed formation, but most of the better known MACBOX genes are the ones implicated in flower organ identity, or known as the ABC genes. I'm going to focus on the first stages: root development, flowering transition, and the meristem identity and maintenance, both in root and in shoot. So, by reverse genetics, we isolate 3 mutant alleles of the Chantal1 or AGL12 gene, and as you can see, all the alleles have a short root phenotype compared to the wild types. Yeah, 2 of them they were in the Columbia background, and 1 of them was in the Lancer erecta background. And 30% of the plants show not well-formed organizing center that in root it calls the chiastic center. And you can see here it has a periclinial division that it should not be in the wild type. And also they have a different pattern in the columella cells. But as I told you, this happens in 1/3 of the plant, so this could not explain the short root phenotype. So we made a quantitative analysis, cell quantitative analysis, and we found that the meristem length were shorter in all 3 alleles. The cell production rate was also lower, and as a consequence, we found that in 2 of the strongest alleles, they have a longer cell cycle. So, because it seems that it was affected the proliferation in these mutants, we analyzed the expression of some cell cycle components, and as you can see here, for the cyclin B marker with Gauss tinction, in the mutant there were less spots compared to the wild type, and in the overexpression line there were more spots. And this was also corroborated by the meristematic cell number, that was less cells in the mutant, and they have more cells in the meristem in the overexpression lines. We also analyzed by RT-PCR some other components, 7 of them, of the cell cycle, and we found that only 3 of them they were reduced in the mutant, suggesting that some checkpoints in the cell cycle were affected, but not all the cell cycle. So in the root, you can find 3 zones: the proliferation, the elongation zone, and the differential zone, and they are characterized by here is the mitosis. Then in the elongation zone, the cells stop dividing and they expand, and in the differential zone, they get the final size and also their special characteristic. For example, the appearance of root hairs in some of the epidermal cells. And it is known that there is a correlation of getting into endocycle. So the differentiated cells start elongating because they have this endocycle and not the normal mitotic cycle. And as you can see here, that using this marker, the root hairs, the root hairs were closer to the QC in the mutant, and it was farther away in the overexpression line that is quantified in that graph. And that was expected because we have a shorter meristem in the mutant, but what we didn't expect is that the root hairs were longer also in the mutant and shorter in the overexpression line, suggesting that Chantal-1 is also implicated in this growing of the root hairs. And more importantly, when the cell size was analyzed in all from the QC cells to the first root hair primordia, you can see that the cells in the mutant started elongated before the wild type prematurely, but they stop growing also before they get to the differentiation zone. That as I told you, in the wild type, the cells are still growing after this point. So we thought that maybe since it is a correlation in endocycle and in cell expansion, this mutant could be also affected in the endocycle. So we measure by flow cytometry the frequency of the nuclei that have higher levels of ploidy, and we have a tendency to find that there was a less proportion of the cells with less 16C ploidy. So I don't know. Okay, so now I'm going to move to the shoot apical meristem. So flowering transition is a very important developmental regulation process, and in this happens that the shoot apical meristem that in the vegetative phase produce only rosette leaves, it is transformed to during flowering transition is transformed to the inflorescence meristem, and on the flanks of this inflorescence meristem, the flower meristems are formed. After that, sometime later, this flower meristem starts to differentiate into the flower organs. But this process is very important, and because the inflorescence meristem stays in the meristematic way until the plant dies, while the flower meristem is a determinate meristem that the stem cell niche is consumed or exhausted after the organs are formed. So it is very important that the identities of these two meristems are make this transition in flowering, the flowering transition is a very, very big network that it has been shown that about 300 genes participate in this flowering transition, so I cannot go to all of them. And also there are 7 flowers described to participate, both environmental signals and endogenous and physiological signals that they have to be integrated in this network. So I'm going to focus only on 2 of those signals. That there's because Arabidopsis flowers during spring and summer. There's the seasons where the photoperiod is a long day photoperiod, and also where warmer temperatures stands, like 27 degrees. And the optimum for Arabidopsis to grow is 22 degrees, but in this case, the warmer temperatures induce flowering. So there are two proteins. There are small proteins that belong to the phosphatidylethylamine binding protein family. That they are terminal flower, TL31, and FT. It is very interesting because these proteins can travel in the plant, and the TL1 is expressed only in the inflorescence meristem, and it can travel to the rest of the meristem, and it represses flowering transition. And FT is the homologous protein, but it is expressed in the leaves, is translated in the leaves, and then it travels to the flower meristem. And this one induce flowering, this one repress flowering. Okay? So to do it, they have to bind to a transcription factor, and in this case it's FD. And when FT binds to FD, displaces TL1, and in this case it has a higher affinity, and it regulates the flowering genes. And among them are the leafy and the apetala one. They give the identity to the flower meristem, and this particular AP1 represses the expression of TL1 here, and TL1 represses the expression of AP1 here. So that's how the identity of both meristems is given. So, returning to our genes, we know that Chantal1 is expressed both in the inflorescence meristem and in the flower meristems, and compared to the leaves, it is higher expressed in the apex. And what we found now is that the Chantal1 is induced by light. In this experiment, we grew the plants 13 days in long day photoperiod, then we passed the plant two days in the dark, and then we turn on the light again for 10 hours. And as you can see, the expression of Chantal1 peak between 2 and 4 hours, and then declines. While the plants that were kept for these 10 hours in the dark, they could not be induced Chantal1. And plants that were for 8 hours in the light, but then we put it in the dark for 2 hours more, it declines faster. So in agreement with this, we found that the red light photoreceptor, that there is phytochrom B, induced Chantal1 because in the mutant of Phy1B, the expression is lower than in the wild type, and this does not happen with phytochrom A or with the cryptochromes. So here you can see very nicely that Chantal1 is late flowering compared to the wild type, and in the mutant, when we analyze the Chantal1 FD and the double mutant, in long day at 22 degrees, the double mutant behaves exactly as the FD, meaning that FD is epistatic with over Chantal 1, and that means also that they are in the same pathway. But when we grew it in short day at 27 degrees, they have a synergistic effect in the double mutant, meaning that they work independently in this condition. So under long day photoperiod, Chantal 1 regulates FD, and we also made chip analysis, and we found that Chantal 1 binds to the regulatory regions of D in 3 fragments, DNA fragments. And about Chantal 2, it's also expressed first in the incipient flower meristem, then it appears also in the inflorescence meristem, and in this case, Chantal 2 is not regulated by the environmental conditions, but the internal conditions of the plant. And here you can see that Chantal 2 is induced by gibberellins, but for this, they need the SPL genes, that they are the squamous binding protein-like proteins, that they are participated in the aging pathway. And they cannot, the Chantal 2 is not induced when these genes are mutated. And same wise, Chantal 2 is needed for the induction of some flowering important genes like SOF1 and leafy. And also, where here you can see the phenotypes in flowering transition. The mutant is late flowering, the overexpression is early flowering, and this overexpression line is early flowering independently of the photoperiod that you grow it. And here you can see that it is very important to induce AP1, and the petala1 gene, and Chantal 2 also regulates it. And here you can see that in the overexpression line, it is very highly regulated, and it is early expressed compared to the wild type genetics. So interestingly, when we observe the phenotype of these overexpression line flowers, you can see that the sepals were very, very big, that they cover the petals and the stamens, and the morphology of these sepals looks more to the leaf than to the sepals, the wild-type sepals. And when you grow these plants in short-day condition, then the carpel elongates and complete inflorescence can grow from inside the carpels. You can see here that the wild-type only have the ovules distribution, and in these strange carpels, they have all kind of the developmental stages. Here you can see that is in this way a meristem in stage 3 where sepals are formed, and they are mixed with mature pollen grains. So to do that, we feel that the plant should be inducing TL1 because it gives the inflorescence meristem identity, and that was the case. Here you can see that in the mutant is down-regulated, and in the overexpression line is up-regulated. We also made a chip analysis, and we found that yes, directly, indeed, Chantal 2 regulates TL1, but this was a counterintuitive regulation because, as I told you, AP1 represses TL1, so it was like that. We have upregulation of TL1 and we have also upregulation of AP1, but we figured that maybe TL1 was overexpressed compared to AP1, so if we upregulate again, AP1 could complement this phenotype. So we cross the Chantal 2 overexpression line with the overexpression AP1, and that was the case. We partially complement the phenotype. You can see here the big sepals that have this vegetative characteristic, And in here, we the sepals were reduced. You can see now the petals coming up, and they have a trichomes, simple trichomes. So in this way, you can we contribute to the flowering network in which both Chantal 1 and 2 can be regulated by light, high temperature, developmental signals, and also that should be integrated with the signals that comes from the leaves, like long day photoperiod and also high temperature. And there are the conclusions. You can read them because I don't have the time. And just to take home, our results and data from other groups point out that the global warming could induce early flowering in at least some of the plants like Arabidopsis thaliana. Thank you very much. Just acknowledgement of that.
So, thank you. Any questions or good questions while we're doing the next?
Thank you for your talk. Nice. What are the signals? I mean, not when you say environmental signals, what does it mean? I mean, what is the mediator of those for this Chantal?
Yes, the environmental are photoperiod.
Yeah.
That is a long day photoperiod. Normally, that it's regulated by circadian genes. That they are in the leaf mostly, and also temperature that is mostly regulated by phytochrome B.
So that's actually my question is whether if you know which protein or transcription factor induces the expression of Chantal 1 or 2.
No, we don't know.
By this environmental.
Yes, I don't know. We don't know how the signals from, for example, phytochrome B.
Yeah, right.
But we know that phytochrome B is in the nucleus. So we don't know that if it is direct or indirect.
Okay.
May I start? So in the Chantal 1 mutants, you said that the elongation is reduced. But have you looked at the meristem size? The meristem size is exactly the same, or it's just the elongation rate that is reduced?
They have the less cells. They have a less meristematic cells and in the meristem, and I don't know. I don't think that I think that they are more or less the same size, but I'm not quite sure. But they are less cells. Yeah.
Thanks for the talk, Benny. So, is it a problem that the plants flowering earlier, or just they are adapting to the weather change?
Yes. Well, we don't know. I mean, for example, in Arabidopsis, maybe it's not such a big problem because they are not pollinated. But when you have to flower, and also there are also other interactions with animals and bees and everything, maybe they start flowering and the animals are not here, and so we don't know that. Also, the stratification, for example, in a mountain, it has been seen that the plants start growing in another height of the mountain because they continue flowering the same time that it should be. They just changed their place where they are growing. So I think that all these scenarios can happen.
Thank you very much,
Thank
Bere.
you. Sorry.
And well, our next speaker is Kalpana Nanjarreddy. She's from Inés León from the National University. Thank you.
Good morning, everyone. First of all, I would like to thank the organizers and Javier for inviting me to, providing me the platform to share our work. Today I'm going to be talking about the role of the kinase, the SNRK1, in deciding the symbiotic associations in Physiolus vulgaris. Well, the plants as the photosynthetic organisms, sucrose is the primary source of energy. So it's very important to understand how exactly the carbon mobilization happens in the plants during the process of the growth, the development, the reproduction, and also in defense and symbiosis. So we do know many kinases which work as the sensors of sugar, and also they help in maintaining the energy balance in the cell, how the cells are going to function. In this sense, two most important kinases are target of rapamycin and SNRK1. And target of rapamycin and SNRK1 are highly conserved in eukaryotes. TAR is the homologue of mTOR in case of mammals. SNRK1 is the homologue of AMP activated kinase in case of mammals, and sucrose non-fermenting 1 kinase in case of yeast. So as we see here, TOR is generally activated TOR is repressed in the stress conditions, and it upregulates the autophagic processes and helps in the survival and the proliferation of the tissues. And SNRK is heterotrimeric kinase which has alpha subunit, beta, and beta gamma subunit, while the beta gamma subunit is very specific to the plants. And under stress conditions, SNRK1 normally upregulates the catabolic processes, and this helps in maintaining the energy homeostasis. And unlike any other plants, legumes are the plants which have a special ability to get into the symbiotic associations with the rhizobium-mediated nitrogen-fixing symbiosis and arbuscular mycorrhizal mediated symbiosis with the arbuscular mycorrhizal fungi. And both these processes are the energy-consuming processes. So it is a challenge for the plants to decide how exactly the plants are going to accommodate these symbionts in this relationship. And when it comes to the interaction between the SNRK1 and TOR, there has been a lot of studies going on, and in the normal conditions we see that TOR kinase actually influences, because when the TOR kinase is functioning normally, it influences the shoot apical meristem and the root apical meristem growth. And on the other hand, SNRK1 also is involved in influencing the primary root growth. But in case of stress conditions, what happens? TOR is the one that is repressed, and it relocates the sucrose in a way that it supports the primary growth. However, the SNRK1, with the mediation of ABA, it gets liberated from the complex of SNRK1 and SNRK2, and then it represses the primary root growth. On the other hand, in the lateral root formation, we know that SNRK1 and TOR1, both TORC1, both of them do mediate the lateral root formation. So this is a bit of a controversial signaling. Why? Because there have been studies which support that when SNRK1 is overexpressed, this is in case of Rhiza sativa, when they overexpress the SNRK1, they see that the normal growth of the plant is repressed. However, it does give resistance against the necrotrophic pathogens. And in other cases, this is a recent work, and here we see that the SNRK1 in fact influences the lateral root formation that is mediated by ARF signaling. In our case, we decided to see what exactly happens when we overexpress the SNRK1, and to begin with, we checked the promoter expression patterns of SNRK1, and we could clearly see that the promoter is expressing in the root apical meristem, and that's in the lateral root formation, And then we could see that at certain stage, the expression is limited to the base of the lateral root, and later on we can see the expression also in the meristem, very strong expression. And based on this, we did overexpression, and also we knocked down the transcripts of SNEC1, and we could see that when we overexpressed the gene, it did increase the primary root length, and also it increased the lateral root density. However, we didn't see much of a difference when we knocked down the gene. So this is in case of Phaseolus vulgaris, and here we are working with the hairy root system, since we do not have stable transgenics here. Okay, so this is about what we found in Phaseolus vulgaris. And next, coming to the symbiosis part of it, very briefly I would like to introduce to the process of symbiosis. Here legumes are the plants which are capable of getting into this kind of symbiosis, and the entrance of the symbiont is through the root hairs, and the plants produces the flavonoids and the bacteria produces the nod factors. There is a chemical dialogue between the host and the symbiont, and then the symbiont is trapped in the root hair, which is, well, as a response of the presence of the symbiont, the root hairs immediately go through extensive deformations to accommodate the bacteria. There is an incoming bacteria, and then by the process of endocytosis, the bacteria is housed and it enters into the plant tissues, and there is the formation of the growth of a special organ, that is a nodule, to house the incoming symbiont. And in this case, the interchange, the main goal of the symbiosis is for the plant to obtain the nitrogen, and the contrary, the symbiont will be benefited with the carbon. And in the arbuscular mycorrhizal symbiosis, we have the mycorrhizal fungi. Also here we see the chemical dialogue between the symbiont and the host, and here the strigolactones are produced by the plant, and the MIC factors are produced by the mycorrhiza, and then there is the first contact point, that is, it forms the hypopodia, and then the hypopodia enters into the, grows in the mycelia, into the plant tissues, in a way that the mycelia is accommodated in the way intercellular and intercellular, and within the cells we can see that the mycelia gets finely differentiated to form the structures called as arbuscules, and these are the places where there is an exchange of the nutrients that are transported, okay, from the rhizosphere to the plant. So here the major nutrient normally we talk about. Very interesting aspect here, because we know that when we know that SNRK1 is the regulator of the energy status of the cell, it is important to understand when there is such a symbiotic process happening, how exactly do SNRK1 mediate this process. So with that intention we started to work, but in the beginning we have to go back a little bit to understand the the signaling pathway, that is the PI3K signaling pathway, where we find the PI3K, the SNRK1, and also the TOR, which are all interrelated. So initially, one of our colleagues published a work which was about PI3K's role in root nodule symbiosis, and there they found that when PI3K was knocked down, it actually affected the progression of the infection thread in case of rhizobium, and hence it did affect the nitrogen fixation, and also there was a drastic reduction in the number of nodules. So, and also simultaneously it did affect various genes that are related to the autophagy. Why autophagy? Because at the end of all the signaling pathway, it is related to the autophagy. And we, from our group, when I was working as a postdoc, we worked on target of rapamycin, and target of rapamycin in fact photocopied the, phenocopied the phenotype that they got in PI3K. So here also we did see the arresting of the progression of the infection thread. So it was aborted in the root hair cell itself, so there was no progression, and we didn't see any rhizobia in the nodules. There was a drastic reduction in the nodule numbers, and whatever the nodules we could find, because these are the RNAi knockdown plants, so we cannot really say that they work like mutants. So we didn't see any colonization in these nodules, so it did affect. And also we could see that in few of the nodules which were formed, there was a little bit of colonization of the bacteria, but the nitrogen fixation was almost nil, because we did see a very high upregulation of the autophagic processes, and there was an extensive vesicle formation, and hence it did not allow the nitrogen fixation. That's what we believe. And from there, we wanted to also look into the part of mycorrhizal symbiosis, and later on we worked on the mycorrhizal symbiosis in the similar conditions of target of rapamycin knockdown conditions. And there we could see that in comparison to, this is the arbuscule of the mycorrhiza, and in control plants, obviously we could see well-formed arbuscules, and when we knocked down the expression of TAR, we could see the stunted arbuscules, are the arbuscule differentiation was affected to a greater extent, and hence we did call them as the abortion of arbuscules. So it was very interesting for us to go from there and to understand what SNEC1 could be doing, okay, in the same symbiotic processes, and hence we went ahead and initially we studied the promoter expression patterns, and we could see that in the initial stages of the infection thread progression, the expression of the SNEC1 promoter was along with the infection thread that is in progression. And in the nodule primordia, in the initial stages, we could see that the expression is in the core of the nodule, but when the nodule reaches to the mature stage, that is 28 days post-inoculation, we could see that the expression was mostly limited to the vascular tissues, similarly in the senescent tissues. So that was a good indication, the good point to begin with. And then we went ahead and we did the functional characterization, both under overexpressed condition and in the knockdown condition. And when we looked into the overexpression of SNEC1, we could see extensive response for the presence of the rhizobium because of the extensive root hair deformations that were seen here. And in comparison to the control, the knockdown phenotype was also comparable to the controls, not only in terms of root hair deformations, but also in terms of the phenotype of the infection thread, okay? So if we look into the infection thread phenotype in both RNAi and in control, we can see that this is a so-called normal infection thread progression. Why? Because it's just like a thread that is growing down, and later on it will enter into the cortical cells, which are going to differentiate. So that's a smooth progression, okay? But here what's happening, I don't say that this is not a smooth progression, this is also a smooth progression, but what we could see here is the infection threads were very, very thick. So there was something happening here, and also when we looked into the numbers of the infection, the events of infection, there was a drastic increase, and that obviously resulted in the increase of the total nodule numbers when we overexpress the gene, but we did not see much of a difference when we knocked down the gene, and it was almost similar. And also we could see in the increased number of the nodules, there was a range of the nodule sizes. Here I'm showing only the mature nodules. So we could see some nodules which are very big and some nodules which are smaller and very tiny. But when we looked into the anatomy of these nodules, there was not much of a difference. They all looked normal. So when we looked into their nitrogen fixing capacities, we did see a very big increase in the nitrogenous activity that represents the quantity of nitrogen that was being fixed, and also we localized the protein and we found it in the plasma membrane and in the nucleus. And next, going to the symbiosis with mycorrhiza, we did see that there was a response for the presence of the mycorrhiza. However, the differences that we could notice is in the first contact of the mycorrhiza with the plant tissues, where we see that the phenotype of the hypopodia is comparable to the phenotype of the infection thread, because here also we can see that the hypopodia is very, very broad. So that is, in some cases, we can say that it is like a resistance response from the plant. So we checked into the marker genes of the AM symbiosis, and specifically we were interested to look at the STR1 and STR2, which are representative of the arbuscule phenotype. And when we looked into the arbuscule phenotype here, we did see that when we overexpressed the SNEC1, there was exactly the same phenotype what we found in TOR. So here also we see that there are arbuscule abortions happening, so it resulted in the development of stunted arbuscules. On the contrary, we saw that there was an increase in the vesicle numbers. And we are yet to analyze some other factors here. We are in the process of studying them, but in the RNAi plants, we didn't see much of a difference. So going ahead from there, we decided to go for, we decided to understand the fundamental basis of what exactly is going on, and we did RNA-seq analysis. And due to some constrictions, we could not do it in the RNA, and also because we didn't have much of a phenotype there, we only went ahead with the the SNEC1 overexpression alone, SNEC1 overexpression under rhizobial symbiosis and under mycorrhizal symbiosis, and we did see big number of genes that have been elevated in the root nodule symbiosis. And there was a big downregulation, a huge number of genes were downregulated in the arbuscular mycorrhizal symbiosis. And when we did the KIG pathway analysis, in the overexpression conditions alone, we did see a lot of genes that were upregulated, that are involved in the biosynthesis of secondary metabolites. But what was interesting to us to see in the symbiotic conditions is the regulation of the genes that are related to the metabolic pathways, and also the ones that are related to the zeatin biosynthesis and nitrogen metabolism. But when we look into the arbuscular mycorrhizal symbiosis, we did see a lot of genes that are related to the flavonoid biosynthesis that were being upregulated. So this is the analysis is under progress, so we are yet to find out which are the particular genes that are responsible for this kind of a phenotype that we are seeing here. And finally, we went on to see the protein interactions. And for now, I have results only for the protein interactions under the rhizobial conditions. And here we could see many proteins that were interacting under rhizobial symbiotic conditions. Some of the proteins of our importance is specifically the allantoinate deaminase and transcription factor BHLH61. Currently we are working with the allantoinate deaminase to understand how exactly it is coordinating, it is interacting with SNEC1 to give whatever the phenotype that we are looking into. Very briefly, what exactly does allantoinate deaminase does is it is a very important enzyme that is involved in the liberation of the ammonia from allantoin. Okay, so this we are looking into the infected cell, and this is an uninfected cell. In the process of nitrogen fixation, once the ammonia is liberated from the symbiosomes, it moves into the uninfected cells where we find it as allantoin, and it has to be converted to allantoinate for it to be transported through the vascular system, and then to reach to all parts of the plants to provide the plants as the required source of nitrogen. So this was a very interesting observation for us, and we have a hypothesis, as you can see, that we didn't come to the end of our work, though it has taken quite a long time for us to have some progress in the work due to many problems. And fine, yes. So here we are here, and I think that the regulation between SNEC1 and the biological nitrogen fixation symbiosis is actually being regulated in this stage where there is a coordination, or it has to be a coordination between the concentration of nitrate and the carbon. And this is the group. I would like to thank my students, Carolina and Gabriel, who was an exchange student from University of Sevilla. And thank you so much for your time. Sorry.
Thank you, Kalpana. Any questions for well, one question because we're running out of time.
Thank you, Kalpana. That was very nice. So when you overexpress the genes, I can see that you have this, you said the infection thread is thicker or is wider, But do you see any other phenotype, like if the cortical cells divisions is delayed, or this is like different compared to the control condition?
No, in fact, we did not see any difference because, as I said, we didn't see any problem in the progression of the infection thread. It was just that the phenotype of the infection thread was different.
I see. Yeah, and the other thing is the nodule size is exactly the same? You don't see, no?
No,
Yeah, yeah.
because as I said, we saw a range of nodule sizes. Biggest nodules that we found were double the size of the control nodules, and then we did see the same size as controls. So there was, I could classify them as 4 different sizes.
Yeah.
And when I say that, we are talking about all of them are mature
Yeah,
nodules.
that means that probably you have like, probably you are interfering also with the regulation, the
Exactly.
auto-regulation of the
Auto-regulation.
regulation, because you have later
Yes.
infection process, and that's why you have
Yes.
this explanation.
Yes.
Thank you.
Thank you very much. Oh, thank you, Calpano. And our next speaker is Roberto Ruiz Pedrano from Simba from the National Polytechnic Institute of Mexico. So thank you.
Good acoustic. Good acoustics here, so we are going with that mic. So, well, I'm gonna talk about a very brief overview of long distance transport of RNA and plants. But first, I'd like to thank the organizers for very kind invitation, especially Javier. So, well, there's a lot of information, very brief information on the intercellular transport of RNAs and proteins between neighboring cells. So, for example, viruses, there's an obvious example, NOD1, short group. And also, we know...
Maybe you need the mic.
Oh, yeah.
No, sorry, yeah.
That's it.
No.
Okay. Okay, so can you hear me? Okay. Okay. So there is also much information about RNAs that move long distance with flow sample collection and aphids thylectomy. May occur at massive scale across graft unions, but maybe not as massive as initially thought. And now we know that there's a lot of exchange, RNA and protein exchange between whole parasitic plants and their host. And most of these RNA, the function is not known clearly. So the phloem distributes long-range signals, that's what we know. That these signals are involved in flower induction, systemic acquired resistance, gene silencing, response to water deficit, and so on. And we're interested in these signals, the proteins and RNAs that might be involved in some of these processes. Okay. So one of the RNAs that we analyzed first, that we isolated from the phloem sample of pumpkin, was this RNA that codes for a transcription factor, and it meets several criteria to consider it as a long-range signal. The first one, I mean, you can find this RNA in the phloem translocation string. Then you can find this RNA moving across a graft union from pumpkin to cucumber, and the RNA localized to extrafascicular phloem, and also this RNA exits the phloem into the shoot apical meristem of the heterograft. So it means it's moving long distance and it's entering, it's exiting that phloem. And well, these are when you analyze the localization of these RNA with more detail, you can see that it localized to the sibilants, the mature sibilants, and obviously you remember that the sibilants lack nuclei, lack ribosomes, or at least functional ribosomes. So these RNA must be moving long distance. It doesn't accumulate in the sibilants. And interestingly, the protein accumulates in the nuclei. It doesn't move long distance, at least in pumpkin. And this RNA is member of a very large gene family, which includes several gene elements in Arabidopsis. And so here's a clade which groups with CMAP1, which is, let me see, it's here, okay? So we have analyzed these homologs in Arabidopsis. And we found that when they are overexpressed, they induce senescence, as you can see here. And interestingly, when we overexpress a promoter construct, which includes the 5' UTR. It also induces senescence. And interestingly, this concept also induces the endogenous gene. So it means that this process is also regulated at the RNA level. Another RNA that we, well, protein, included by this RNA, that we produces a non-force of the cellular signal, we found this moving long distance in the phloem is the translationally controlled tumor protein, which is almost ubiquitous in eukaryotes. And well, we found it initially in the phloem sac of pumpkin, but we found that it's also enteral phloem sac, telegraphed holoparasite transcriptomes and proteomes. And this is a very interesting protein because it, for timing it has been documented that this gene regulates cell size, organ size, and also cell number through this potential G protein binding pocket, which will be important later. And well, we know that TCTP in pumpkin, phloem sac, it promotes proliferation and also increasing in cell size, and it accumulates to very high levels in the phloem sac, and is also induced by viral infection. And as you can see here, it accumulates to very high levels in the apex, and also in the C elements, mature C elements that, remember, they do not have nucleic ribosomes. And interestingly, the protein is barely detectable in the apex. So it could mean that the RNA itself is involved in this process in cell proliferation. Now, there are two genes in arabidosis, two TCTP genes, and they must have different functions because when you knock out one, the other cannot compensate for the function of the lost gene. And we have analyzed this gene, and when it's expressed in tobacco, it moves long distance and it induces aerial roots in the graft germ. And this correlates with the localization of this protein in the nuclei in the cyan, in the non-GM side. And well, this movement, this RNA movement is bidirectional, as you can see here. When you have, let's say, the GM and the wild type, the RNA moves in that direction, but also in the opposite direction. So it means it doesn't move necessarily through the phloem all the time. It must move also through plasmodesmata, through neighboring cells. Now, an interesting fact is that this protein, and also the pumpkin phloem protein, TCTP, induce plant regeneration or organogenesis, but not TCTP1. So that also underlies the different division of labor of these genes. And well, they have very similar structure. The sequence is almost the same, except for this deletion. And in fact, when we introduce this sequence, the TCTP inserted into TCTP2, it behaves more like TCTP1. So in terms of regeneration efficiency, so it decreases regeneration efficiency, and the opposite happens here with TCTP1. When you delete this sequence, it behaves more like TCTP2. So it increases its regeneration efficiency. Now, I mean, you are going to have a hard time trying to establish the phylogenetic relationships of this gene based only on the sequence, at least in plants. But interestingly, if you analyze the predicted 3D structure of this protein, it falls neatly into two groups. One which contains all the fungal TCTP, the human TCTP, TCTP1, and the other one which includes, for example, many parasites, for example, Plasmodium TCTP, and these TCTPs, TCTP2, CFTCTP, and the potato TCTP. So we have proposed that maybe TCTP, the potato TCTP, behaves as the other TCTPs in this one regeneration. So we have agroinfiltrated these potato plants with these constructs, and we found that the RNA also moves long distance to the apex, and well, as you can see, to the apex and to the roots. And interestingly, when it moves to the roots, it induces formation of immature tumors compared to the control. And it also induces the generation of plantlets, stones and plantlets in the roots. So it means it behaves like TCTP2, this product. So these proteins, these RNAs must be moving as ribonucleic protein complexes, and it has been shown for some of them that one of the most important proteins that bind RNA in the phloem is the 16-base phloem protein, and it moves long distance, it localizes to the seed element, and also it's able to bind RNA in a non-sequence specific manner. It binds the RNA and moves its own RNA and other RNAs to neighboring cells, and it also moves RNAs long distance. And in fact, when you overexpress changeably this protein in pumpkin, it increases drum tolerance very significantly. So what's the function of this protein? Well, we think that this protein and this whole system of RNA movement in the plant is involved in the response to stress. In fact, this gene was described just a database as something that is induced by elicitors. So we think it's involved in response to stress, to biotic and biotic stress, through the transport of RNAs, which are later translated in these cell tissues. And since we're in a biotechnology department, we like to look for application for our work. And well, this is an example. This is citrus greening or vine bean, which is a disease which is devastating citrus worldwide. So it's caused by a bacteria which accumulates in the seed elements in the infected plant. And the first symptoms are the yellowing of the leaves, asymmetric yellowing, poor fruit quality. And then, well, the most important aspect is that it eventually kills the plant. There are no treatment, there's no cure for this disease. So we thought we might deliver these antimicrobials to the seed elements to try to control this disease. So the idea is quite simple. It's basically fuse this protein to antimicrobials. And we have to consider here that these are the antimicrobials, they can't enter the phloem, or for example antibiotics, because the phloem is isolated in plasmid domain. So for example, you express something in the mesophil, it doesn't necessarily move a protein, for example, or RNA move into the phloem. So if we fuse these proteins to these antimicrobial proteins, for example, a resident phloem protein, so in principle we would reach the phloem. And so that's what we did. We transiently expressed these proteins to mitigate this disease. And the interesting point is that we use diseased plants. So these plants were already with a lot of symptoms, loss of bacteria. And after 100 days of treatment, the plants recovered. And the amount of live bacteria in these plants was very low or undetectable. And finally, when we did an open field trial of these Mexican light stably expressing this, that citrus equivalent of PP16, we found that the plants were, well, the disease was mitigated, the plants looked productive and more or less healthy, and more important, the control plants were all long dead. So it means this is a strategy that can protect plants from pathogens that invade the vascular tissue. These are the conclusions. Well, there's a long distance signaling plant rely on RNA proteins and many other molecules, and it relates several processes, that some of which have been described here in detail. And these resident phloem proteins can be used to deliver targets of interest to systemic tissues. And I would like to thank again the organizers and all the people involved in this work, present and past collaborators. Thank you very much.
Hi, nice talk. I'm wondering whether these proteins that are traveling through the phloem, they are freely traveling, or the traveling is modulated by some kind of signal?
That's a very good question. I mean, that's a very good question. It seems that many of these proteins are traveling as complexes. And in fact, these proteins, the population of these proteins from the phloem transportation stream is not constant. Because if you, by AV stylectomy, you detect the phloem proteins from different parts of the plant, you'll see different populations. So it seems that they are constantly entering and exiting the phloem. And in fact, there is who would like to say it's like the equivalent of blood-brain barrier underneath the meristem. So not all proteins can enter the some proteins cannot enter the pseudo apex. Some of them can, and some of them can transport RNAs. For example, some RNAs can move into the pseudo apex by, as we have seen, for example, PP16, but some others cannot. So yeah, it seems that it's a very tightly regulated process.
Thank you, Roberto. So the movement of the resident proteins, the one that you were saying that you can put the fusion with the antimicrobials. So is it moving in both directions, or can you just go in one direction?
In that case, it moves also through this.
Okay, so it goes. Can you get like the proteins or the fusion protein accumulated in one specific area, or is homogeneously?
Well, we have to do that. I mean, we have to analyze that. We really don't know. I mean, when we, for example, in the case of the agrobacterium transient expression, let's say we apply that to the stem, and we can detect it in, let's say, more active tissues. But in that case, we didn't analyze the roots of the plant.
I'm curious if you have, for instance, like an infection in some part of the shoot or the leaf, you will have an increase of the transported proteins in other places. So it's required.
I don't know. I mean, that's a good question. There's a lot that we don't know about this system. That's a good question.
We have a question. I always wonder, it's a very basic question, like what would be like this evolutionary advantage or something that the signal would be RNA if it's such a labeled molecule and everything. So what's not a protein? So how is it possible that it's not received the signal in the other cells, in the target cells?
That's another good question. I mean, we know that some of these components are present also in non-vascular plants, for example. In fact, the PP16 gene, it doesn't exist for example, in chlorophytes. There's only the extended synaptotymine. In fact, the PP16 is just the C2 domain of the extended synaptotymine. So it seems that the machinery is there. Now, what about the RNAs? I mean, why is the RNA a signal? Well, in some cases, it's very complex. But in some cases, you find both the RNA and the protein moving through the whole plant. But for example, for CMNAP-P1, it's just the RNA. In other cases, at least we find for the TCTP, the RNA and protein are both in the phloem cell. But the interesting thing is that, well, it may be the coverage of these transcriptors and proteins is not very complete, but it seems that in some cases, it's just a protein there, but not the RNA. But depending on the species, for the herbaceous species have a more limited phloem count.
Thank you. Thank you very much, Roberto. And we're having some technical issues. These issues are always present in these conferences. So our next speaker, Ariel Hernandez, he's a PhD student, so he's here from Institute of Technology from the National University. So thank you very much, Ariel.
The microphone.
Can you hear me well without the microphone, or?
It's better if you use.
Okay.
Kind of.
First first I'd like to thank the organizers for the opportunity to present at this conference, and especially thanks to Javier. And well, I am a PhD student in Dr. Patricia Leon's lab, and my work is on the ortholog of the GLK or glycan transcriptional factors in mercantile polymorpha, and its role in chloroplast development as well as in plant vegetative development.
Time is running out. Eso es lo que estoy haciendo.
¿Quieres mi computadora?
Or we can skip the next one. Maybe no. So, sí, no importa, pero para no perder tiempo, because we're over time, so. A ver si pueden cambiar.
Bueno, no, esa no.
Ya está.
Ahí está.
Ya está. Vamos a que el computador está
Bien.
con.
Check it out.
Ya. Pues el doctor puso una azul.
So because of the technical issues, we're going to change the order of the talks. And so the next speaker So our next speaker is a very nice guy, me. I also want to thank the organizers from the Congress that invited us. And I'm going to talk to you about protein secretion in cell plants, which is one of the topics I've been working in the lab lately. So, well, I want to start saying that, well, angiosperms are the most diverse group of land plants, as we heard also in the early talk this morning by Noni. And one of the reasons maybe is this successful evolutionary diversity is the flower, which is a reproductive structure of angiosperms. And in probably contradictory terms, we found that most of angiosperms are hermaphrodites, which means that they possess both female and male sexual organs in the same structure in the flower. So this would lead us to think that self-pollination, inbreeding would be the rule in these plants, which would contradict to this diversification in the land plants. But no, actually, plants also know that the incest is a very big taboo, as we do. So there are many strategies, as also we heard it earlier in this morning in the plenary session, that there are some barriers that prevent self-fertilization. The strongest ones are those that are genetically regulated, which is self-incompatibility. So in the morning we heard about the Papa system. We're gonna hear now about Nicotiana, but just in the surface. So this is a discriminatory system that recognizes and discriminates between self-pollen to inhibit it and cross-pollination to promote this fertilization. So this one is genetically related by the S locus, where the male as a determinant is inside, is located, and is being expressed especially in the male organs, and the female as a determinant in the female organs. In the case of Nicotiana, different to the one in papaver, as we heard earlier this morning, the female as a determinant is an sRNase, where is a ribonuclease, which is called sRNase, and the male as a determinant is SLF, which is an FBUX protein. So what we can see in both incompatible and compatible crosses is that pollen grains germinate, but only in the compatible crosses, pollen tubes will grow happily towards the ovary. This is actually a very quite complex system, where vacuoles and many cellular processes are involved. But our next speaker, Felipe, will be talking about this. But what I can tell you is that in both cases, sRNases that are secreted in the style are incorporated in the pollen tube. And in the incompatible process, these vacuoles are disintegrated and the sRNases are released. So they have this cytotoxic effect on pollen tubes. And you will see that why this is important. So this also means that there are other genes that are involved and they are not linked to the S locus. One of these is NHRXH. This is a pterodoxin, which was identified to be involved in the cell system. Pterodoxins are proteins that are conserved from prokaryotes to eukaryotes, but in plants it's the only group that they are quite diversified. We can find at least 20 genes in Arabidopsis thaliana genome, and there are so many groups, and they are matched with their localization, either in the mitochondria or the chloroplast, etc. The H group, where NHRXH belongs, it was originally described as a cytosolic group of proteins. However, through the time they have been related to the ER, to the mitochondria, and one of them is secreted, which is the case of NHRXH, which is secreted to the extracellular matrix of the cell. So this could lead us also about its role in self-incompatibility. So we have worked in both cases, in both ways, about the role, but also about this weird cellular localization. So just about the role, I'm going to say this. As I told you, sRNases, but also NHRXH, which are in the style, they are incorporated into the pollen tube. So for pollen rejection, it is needed that the sRNase being reduced by the NHRXH. It happens in only one disulfide of this enzyme, and the result is that the activity of ribonuclease increases 7 times. Without this increase, there is no pollen rejection. That's the role of NHRXH, the biochemical role. So, but talking about cell localization, which is this talk about, and analyzing the structure, well, Felipe's lab got the structure of the protein, which is a typical thyroidoxin structure, but this group 2 also is characterized by having extensions towards the end and or C-terminal. The N-terminal of NHRXH has a prediction of a cleavage site between the 16th and 17th residue, which could give us a hint about its localization. So we proved that it is not because of this tissue is quite secretory, and it's not the result of that. By transient expression and size, we confirmed that the protein has information for its secretion, besides not having a clear signal peptide. So to identify where the information was, we made some deletions, and here and so on. I will show you experiments in onion epidermal cells by expression transient assays. So we found that yes, it is in the N extension that where the information for its secretion is located, but surprisingly, it's not the alpha domain, the one that was predicted as a signal peptide. It is in the beta domain. When it is deleted, it is this 11 residue long, the one responsible for it. Interestingly, when we fuse that beta domain or beta motif, it is sufficient to secrete or lead secretion of GFP. So it is the responsible one. Analyzing this sequence, it was even more surprising and more weird, because it's quite hydrophilic. The opposite of what would we expect from a signal peptide. Because signal peptides needs to be hydrophobic to be recognized by SRP. This is SRP, which is, well, that's a unit M from SRP, which is a component that recognizes the signal peptides while the protein is translated, and in a co-translational event, it is then translocated to the ER where the translation ends. So it goes to the ER, to Golgi, and secretory vesicles. So the physicochemical characteristics of the M beta tells us that it cannot be recognized by the SRP. So how it could reach the extracellular space? So first we have to follow what was the route it was following. So we added the endoplasmic reticulum retention signal to both NHRXH, the full protein fused to GFP, and only the fusion with the M beta motif. So in both cases, we found that it was retained in the ER. So it uses the ER. When RNA cells were treated with refundin A, secretion was also abolished. It means that it goes through the Golgi. So finally, by immunolocalization assays, we found that NHRX8 was located associated to these vesicles, which presumably is just in the style, of Nicotiana lacta. So presumably they are secretory vesicles. So although it is not a signal peptide, it follows the typical or the classical pathway system. So Andrea in the lab tried to characterize this peptide, and he found that it was present in other proteins, particularly for animals, but also for plants and fungi. This is a consensus sequence, and we classify them according to the information regarding its localization. If they are secreted, if they are not secreted, if it is questionable, and if nothing is known about it. So first, he defined that the first three residues, which are the most variable ones, are not necessary or required for M beta to function as a signal sequence. And then comparing the beta motifs from secretory proteins with those that are not secreted, we found some positions that are interesting. First, this one, which is completely conserved in the secretory. We replaced them from the glycine that was found here, but secretion was not abolished. But the ninth position, which is a serine residue in all the ones, and sometimes we can find in alanine, yes, secretion was abolished. So it's required to have there a neutral residue to maintain this function. Something interesting was the 8th position, because serine was found in NHRXH, and also in some plant pterodoxins in this area that nothing is known. So the glutamic acid was mainly present, but all these are from animal proteins. And aspartic, which is only one carbon different of size, it was in the non-secretory ones. So when we replaced the serine for aspartic, yes, secretion was abolished. So this serine is required for secretion. But surprisingly, when we replaced it for glutamic, secretion was also abolished. So we have two possible scenarios. One is that maybe a glutamic residue would be required for animal proteins to work as a traffic motive. We don't know. But the other one is that maybe this pathway is exclusive for plant cells, and we are trying to work on this. If we change it for a hydrophobic residue, it doesn't happen anything, which is the last one. So, looking for different pterodoxins that were in that gray area, we found very interesting groups. One is a purple one, that if you won't see, but I will tell you, there is a glutamic residue in that position that we changed. And interestingly, there are groups with this pterodoxin that it is known that is localized in the mitochondria. The yellow group is grouped with NHRSH, so we could predict that the rest of these proteins can be secreted. So this motif, or analyzing this motif, could lead us to predict protein localization. And finally, this group, which is only one protein, has an alanine in position 9, so we can predict it is cytosolic. So this is a very interesting thing, because also there is a a huge discussion about how and why are so diverse these proteins in plant cells, and how could they be, if they are promiscuous or not, towards the target protein. So changing just a few residues in this extension could give not only specificity, as we know it happens with NHRXH, towards the target, but also to the cellular localization. So changing this, you change your ticket, so you can go to mitochondria or going out or whatever. There are new tickets now, e-tickets. We also proved if it was in which position we are, if it's important, all secretory or predicted secretory proteins have the N-beta motif towards the N-terminal. So we decided to assess the position as well. If it's in the middle of the protein, doing this chimeric one, this is NHRXH without the N-terminal, then N-beta and then the GFP, we found that it is not secreted. So if it's in the middle of the protein or hidden, it won't work as it. We would expect that being in the carboxy terminal, it wouldn't be secreted, but it was secreted. This was surprisingly when we had to generate more questions. But if we see the structure of GFP, we can see that the N-beta motif towards the N or the C would be exposed and free to the solvent. So in the very similar way as it happens in NHRXH. So this motif has to be a disordered thing and free to the solvent to be recognized, but something that now we are trying to catch that would lead it to the secretory pathway. We also inverted the sequence, and it worked as well. So it is also the charge of the motif itself, because it's, I forgot to tell, it's almost like a palindrome. So these positions are important for the charge of it, not the sequence itself. It's not that rigid. And well, if we know that secretion occurs in the N-terminal and it goes through the ER, what happens when it's in the C-terminal? We would think that it would skip it or bypass it. But no, we also added the retention signal to the ER, and it was found to be retained in the ER. So this means that it's a different kind of entrance to the ER. This is not a sector peptide. This is a transit peptide to the ER. Why? Because the protein needs to be translated, fully translated in the cytosol. So if the unknown element recognizes it, it takes it to the ER and follows the way. So what we are suggesting is that this semi-conventional, because it's using the conventional pathway, but the first step is different. And it's not an unconventional one because it's not bypassing any of these elements. So it is the initial point, and we are after that now. We're working on that. But it's like, well, we make the analogy like transcription factors that are in the cytosol. And once the signal is received, they expose the nuclear transit peptide and they go to the nucleus. So maybe that could happen here in plant cells. In this case in particular, in the styles, the protein would be secreted. It will be integrated into the pollen tube where it takes it function, reducing sRNAs. This motif, the N-terminal motif, is also required to interact with sRNAs. So the motif would be hidden and masked. So that's how NHRXH would be trapped in the cytosol and would be reducing all the NHRXH. So this could lead us also to understand how it got into the pollen tube. It stays into the pollen tube and it's quite specific to this enzyme. So that's the work we're doing in the lab. We're actually trying to have the real structure. This is an in silico model, and that's what we're doing in the lab. So I want to thank, of course, the lab, the students in my lab, who have, well, some of them are not there anymore. And Andre is doing his PhD and working on that. Rodrigo is starting to having this complex I'm talking about. Felipe Cruz, who is one of my biggest collaborators and helpers, actually I was born in his lab. And well, everybody, so thank you very much.
that, do you know if that, that region is phosphorylated or not?
No, we haven't tried that. Yes, it would be interesting to try that and maybe like a switch to be recognized or not. Yes, actually the settings are very interesting. Yes.
Exactly.
Uh-huh.
Thanks, Javier. So this since goes through all these conventional, semi-conventional pathway, and then it goes to the extracellular. So how is the internalization into the pollen tubes?
Well, Felipe will talk about that in a few minutes, because, well, years before, actually Felipe also found that sRNAs interact with many other cellular components, and that's how it is incorporated within the pollen tube. The process, I will not answer because that's part of Felipe's talk, but I think NHRX8 would come in this package.
Right, but any of these motifs that you just described are not involved in that internalization process. It's just for the secretory pathway.
I have no idea because the evidence we have in vitro is that these motifs are required for interaction. Well, maybe yes, because they're important to interact with sRNAs.
Right.
If we eliminate the N-terminal motif, it doesn't interact with it, but it doesn't reduce it either. But if we delete the C-terminal one, it doesn't interact in a stable manner with sRNAs, but it reduces it. So maybe that's the way it looks like. Se cuelga del tren. Sorry, it's from the train to a right towards the cytoplasm, but I don't know how it happens.
Thanks.
Mhm. No, well, thank you very much. And now our next speaker is Ariel from National University, Institute of Technology. Others.
Well, I just presented myself before, so I think I'm gonna skip that. And well, what I would like to address first are plastids. Plastids are essential organelles in all photosynthetic eukaryotes. And these organelles are pretty interesting because, first of all, they're semi-autonomous and still retain a reduced genome. Also, when we analyze the coevolution of plastids and the organisms that bear them, we'll see that most of the lineages only possess a single plastid subtype associated with the photosynthetic process. However, in land plants and concomitantly with the transition to land and also with a more complex body plan, we have now the emergence of or radiation to several plastid subtypes. And these specific plastid subtypes also perform particular functions in the tissues or organs they reside in. And very importantly, these several plastid subtypes are able to interconvert into one another through communication between the organelle and the cell nucleus. Now, this communication is critical because out of the around 3,000 plastid targeted proteins, most of them are encoded into the nucleus. So we have signals coming back and forth, and we have the nucleus signals are referred to as anterogrid signals, whereas those that come from the plastids are referred to as retrograde signals. Now, we have two subdivisions of retrograde signals. We have operational signaling when they come from already mature plastids, such as chloroplasts, amyloplasts, etc. Whereas the biogenic signals come from developing plastids. And as you can see in this scheme, these signals are involved in different processes. Now, plastid signals are actually very complex and they have a very varied chemical nature. We have metabolic signals and also signals that derive or arise from processes that occur within the plastids, such as plastic translation or plastic gene expression. Now, all of these signals converge and are transduced through signaling cascades into the nucleus, where they modulate the activity or the function of transcriptional factors, such as the golden two-like or GLUT, which are particularly critical for this process because they are not only involved in the transcription of photosynthetic associated nuclear genes or FANGs, but also in the transcription of other transcriptional factors that downstream are necessary for the correct function of the chloroplasts, because they participate in the expression of genes such as those that compose the protein import apparatus or the chlororibosomes. The GLUT are a family of transcriptional factors and are usually present in the form of parallels in most plant models. What I'm presenting here is the case for Arabidopsis thaliana, where there is GLUT1 or GLUT2. And as you can see here, only in the double mutant, we have a chlorotic phenotype. Additionally, when we analyze the ultrastructure of these plastids, of the chloroplasts in the double mutants, the double mutants lack grana, which are the stacking of the thylakoids, unlike what we see in the wall type. Additionally, when the expression of photosynthetic genes is analyzed, we see that the double mutants have a reduced expression of photosynthetic genes, and very importantly, when overexpressing either GLUT1 or GLUT2, we see the differentiation to chloroplasts in tissues that in wall-type plants they are not photosynthetically active, such as in roots. Hence, these transcriptional factors are not only critical for chloroplast maintenance or the photosynthetic apparatus function maintenance, but also directly promote the transition or differentiation to chloroplasts. Now, in order to understand how the function of these transcriptional factors in chloroplast retrograde signaling has evolved, we have to look beyond the tracheophytes or vascular plants. Hence, looking at the bryophytes is critical. Within the bryophytes, we have three lineages: hornworts, liverworts, and noces. But very interestingly, only in liverworts, we have different plastid subtypes, whereas hornworts and noces only have chloroplasts. In Marcantia polymorpha, for example, we have chloramyloplasts in the storage tissue, proplastids in the meristematic tissues, and chloroplasts in the photosynthetic tissue. Hence, this plant would be a feasible model to study the evolution of this transcriptional factor, particularly its function into this process. Hence, with this in mind, we decided to take on the study of the orthologue of the GLUTs in Marcantia polymorpha. And very interestingly, unlike what I just told you before, in Marcantia there is a single orthologue. And also, this single orthologue is pretty particular as compared with those of already characterized GLUTs, because as you can see here, the classical primary structure of a GLUT is a GARP DNA binding domain and a GCT box for homo or heterodimerization. And what we see with the MP GLUT is an additional N-terminus domain, which is a pseudo-response regulator, which is also present in all other GARP family proteins. The GLUTs are sort of inside the GARP transcriptional factor family. So this raises an immediate question: what is this transcriptional factor in this plant doing? Is it actually functioning as a GLUT? Maybe as a PRR2? Or maybe as both? In order to answer this question, we decided to generate mutants and overexpressing lines. And as you can see here, the mutants and overexpressing lines exhibit phenotypes that we would expect in a bona fide clock because the mutants are, for instance, are chlorotic and the overexpressing lines are hyperpigmented. Furthermore, when we analyze the expression of photosynthetic genes, we would see that in the mutant, these are downregulated, whereas in the overexpressing lines, these are upregulated. We also perform ChIP-seq analysis, and some of these genes are direct targets of GLUT. As I told you before, Mercantia already has different classic subtypes, and we can see that through histology. In different tissues, we have different classic subtypes. So we have here in the photosynthetic region with the chloroplasts, and here there would be the chloramyoclasts, which are non-photosynthetically active. And as you can see here in the overexpressing lines, we see now chlorophyll fluorescence in the parenchyma tissue, which is in turn suggesting that MP-GLUT is inducing chloroplast differentiation in the parenchyma cells. Furthermore, when analyzing the ultrastructure of the chloroplasts of these plants, we see that the endomembrane system in the mutants is less complex than that of the wild type, and contrastingly, we see that the overexpressing lines have a denser thylakoid packing. Thus, MP-GLUT is needed for proper thylakoid development. An additional aspect we decided to tackle was the development of the photosynthetic tissue. This because when affecting somehow chloroplast development in eudicots, you see defects also in the development of the photosynthetic tissue. And in Mercantia, the photosynthetic tissue is inside these hollow chambers, which are called air chambers. And as you can see here, the Waltech air chambers have lots of photosynthetic cells, whereas the mutants are basically depleted of this cell population. And in contrast, the overexpressing lines have more cells in these air chambers. Up to this moment, what we can say is that MP-GLUT mutants and overexpressors exhibit classical GLUT-associated phenotypes. Hence, we can conclude that this transcriptional factor in Mercantia is indeed has a bona fide GLUT function, despite the presence of an additional N-terminus. However, and we see other phenotypes that have not associated with a GLUT function in other model systems. For instance, as you can see here, the wall type grows as a bilateral, with a bilateral symmetry. When we see the mutant, we lose this symmetry. The plants are asymmetric. And when analyzing the overexpressing lines, as you can see here, they are smaller than their wall-type counterparts. And when we analyze why is this developmental defect in the overexpressing lines, we see that it is due to a retardation in growth. So it's not only that they are small because the growth is retarded. Furthermore, we decided to analyze the development of dorsal structures through scanning electron microscopy. And as you can see in these panels, the development of the thallus, as seen from above, is clearly affected in the mutants. Additionally, when we analyze the development of the air pores, which are analogous to the stomata, the mutants are unable to develop a full epidermal ring, unlike what happens with the wild-type or the overexpressors. And also, when analyzing the development of the gemmacops, which are a fundamental structure in Mercantia that allows this plant to reproduce asexually, the mutant at 20 days old still lacks this structure. However, the these mutants are able to develop this structure later on in development at 25 days old. We know that for the development of this structure, a transcriptional factor called MPGCAM1 is critical. Because in mutants of this transcriptional factor, there is no GMACAP development. And we know that MPGCAM1 is regulated through two signaling pathways, one dependent on cytokinins and one on the dimer formed by the carotene receptor 2A and the F-box protein MAX2. And what we found was that MP-GLOT seems to be directing influencing the development of this organ through this signaling pathway, because this transcriptional factor is directly regulating MAX2. As we can see here with the expression analysis and also with the chip qPCR analysis, where MP-GLOT is binding to the promoter region of MAX2. So what we can conclude with this work is that MP-GLOT is a bona fide GLOT, despite displaying an additional N-terminus domain on its primary structure, and also that MP-GLOT is necessary for proper thallus development, including the development of dorsal structures such as air pores and GMACAPs, and that MP-GLOT regulates GMACAP development through the MPKI2 MAX2 pathway. And I would like now to thank the people that participated in this work within Patricia Leon's lab, and also the people that helped me with the electron microscopy analysis in the IBT and also in the science school, and to our collaborators and the funding agencies. Thank you so much, and I'm happy to take questions.
Any questions?
Fascinating. And it's funny that the GLK RNA is also in the form of location between Boston and autistic something, so might have something to do with non-cellular autonomous function.
Yeah, well, in Arabidopsis, we know that, in Arabidopsis, it's known that it is non-cell autonomous, but we would have to find out if in your context is also the case.
I need those, but I also has plasmodesmata function.
Yes.
Plasmodesmata,
Of course, it also has postmodesmata.
Thanks, Javier. So I can see that in the overexpressing lines, it's it's greenest, but also it's smaller, isn't it?
Yeah.
How to check if that is just like the cells are smaller or they change the size of the cell?
Actually, the cells are not smaller, but what we think that happens is maybe related to cell wall. I didn't present to you all of the data from the transcriptome because it's a lot. But one of the overrepresented categories is actually cell wall, and we think that might be because it's more constrained. And also we have a lot of genes related to ROS, which might, the plant might be also pretty stressed because of the amount of photo stress.
Right.
And that might also explain the retarded growth of these plants.
Exactly what I was going to ask you, like if those plants are more photosensitive.
Yeah.
And it also looks like the air chamber is also smaller compared to the wild type. Yeah.
In the overexpressing
In the
line?
overexpressing line.
No, actually it is, it's wider.
Okay.
It's wider.
Thanks.
one last question.
So this functional innovation in these large transcription factors is conserving bryophytes and other taxa there.
Well, in terms of chloroplast differentiation, yeah.
But the protein.
Ah. You mean the structure?
The structure, the PR and the.
Yeah.
and the-
The PRR is also present in Salaginella mellendorfi, which is a lycophyte, and also in ferns. So it would suggest that it might have been lost later in evolution.
Okay.
Thank you very much, Ariel. And our next speaker is Felipe Cruz from Chemistry School from the National University of Mexico. Thank you, Felipe.
So thank you very much, Javier, and really appreciate the invitation to talk about my data in this meeting. I'm going to start with some general things about self-incompatibility, but I think that you become expert today about self-incompatibility after the wonderful talk from Nani and Javier. I'm just going to mention something that to be in the context of this talk. So just to remember which are the possible pathways of the gene flu in plants. So you can have here, here this flower, this hermaphrodite, where the male and female organs are close. And this increases the probability to get cell pollination and also cell fertilization. And also, the pollen can come from another flower in the same plant, and that's the same thing. And both gene flux by pollen is going to lead to endogamy and all the problems that come with this. And the plants prefer, in some way, to have cross-pollination with different individuals of the same species. What is the consequence of the inbreeding? Just briefly, the main problem in plants is the loss of fertility. And this is a big problem because there is no generation there is not the next generation, and it's going to be a problem. So Javier and Noni talk a lot about the S locus, where the male and female determinants are localized. And just to remember you, this is the S locus in the self-incompatibility based in sRNAs, because the female determinant encodes for the sRNAs and the male determinant is a complex loci, to say something. This is a set of SLF that are involved in the pollen recognition. They're going to define the pollen rejection response when these interact with the sRNAs. So I'm not going to mention this, but this genetic control is present in very important families like Solanaceae, Rosaceae, and Rutaceae. They have very agricultural important species. So where the flowers reach the self-incompatibility, this is when the flowers accumulate in the pistil a very large amount of the sRNAs, and this is when the flower opens. So in this talk, I'm going to talk more about the sRNAs and the female part. And here you can see this famous picture that everybody presents here, but this is one was taken by Bruce McClure in the University of Missouri in USA. And this is just to show you the anthers and the pistil. And this is one longitudinal slide of the pistil. And you can see here a very thick extracellular matrix. And in this one, the sRNAs are going to be present because this is a secreted protein. And by this extracellular matrix and the transmitting tract, you can see here a pollen tube growing through this, and this is going to get the ovary if this is a compatible pollen tube. So when the pollen tube is traveling around the transmitting tract, you can see here an S10 pistil, and you have here an S10 pollen. So when in this situation, this is an incompatible cross. You can see here the sRNAs in red, and the pollen tube rolling here, and the sRNAs is inside the pollen tube. So contrary, in a compatible pollen tube, you can see also the same thing. Here you can see the same pistil with the same genotype, and you have here the S105 pollen. This is a compatible cross. And also you can see inside the pollen tube the sRNAs. But then the question is how the pollen tube escapes to the cytotoxic activity of the sRNAs. So here I want to show you this, how the pollen tube can solve and escape of the cytotoxic activity of the sRNAs. You have here a pollen tube growing by a pistil, and you can see here in white the cell wall of the pollen tube, and in blue the sRNAs, and 120K protein is, let's say this is labeling this compartment that could be a vacuum. I mean, this is a vacuum. So you can see here in 16 hours after pollination that the sRNAs is contained inside this vacuum, and this is still there after 36 hours. But contrary, in an incompatible cross, the sRNAs is here in this pollen tube. You cannot see clearly the vacuum, and you can see there are some sRNAs. But later, 36 hours after pollination, the sRNAs is dispersed in the whole cytoplasm. It means that these sRNAs could have the cytotoxin activity and degrading RNA. With this model, with this data, we can do a very easy model, just saying that pollen tubes escape in a compatible cross, just maintaining the sRNAs in a vacuum. But it's not, the life is not so easy, and we know that another genes are playing a role in the self-incompatibility biochemical cascade. So there are another genes, we call them modifier genes, at least have been identified in Nicotiana, 5 genes that are playing a role in the pollen rejection response. So we have a model. This is a very, very complicated model. I'm not going to mention you the details, but just the general thing here is in an incompatible cross, the sRNAs are going to go to the vacuum, and if the cross is incompatible, this vacuum is going to disrupt, and the sRNAs are going to be free in the cytoplasm, and they are going to degrade the RNA, and then the pollen tube growth is going to stop. Contrary, in a compatible cross, the vacuum is going to stay stable, containing the sRNA's activity, and the pollen tube is going to grow because the protein synthesis is going to go okay. So then, here the question is what triggers the vacuum containing sRNA disruption in incompatible pollen tubes? This is a problem. As you can see here, this is very complicated. This is occurring in Nicotiana alata. This is a self-incompatible species, but this is a very interesting question. How the pollen tube is going to control that the sRNAs stay inside the vacuum. So to take this question, we are starting to work with another species that naturally is a self-compatible species. This is Nicotiana tabacum. This species doesn't reject the self-pollen because it doesn't have any sRNAs and neither modifier genes. In normal conditions, this species accepts the self-pollen. But if this plant is expressing an sRNAs, this plant rejects the self-pollen. So, and then we try to take this work with classical biochemistry, purified sRNAs from pistils from Nicotiana alata. You can see there, these are very pure fractions. And then we label these sRNAs with dexazred, and we check that the activity it's okay after labeling. And we use these sRNAs in vitro assays to see if this sRNA is going inside the pollen tube. You can see here a pollen tube growing in vitro, and the sRNA is in this case in magenta, and you can observe that the sRNA is very close to the pollen tube along the whole pollen tube, but this is in vitro. But when you do some orthogonal curves here, slides, I mean, you can see the sRNA inside the pollen tube. It means that this thing is working, it's nothing new. And to get a similar environment to the pollen rejection in the pistil, we use now this slice using pistils after pollination, 3 hours pollination. The pollen tubes are going to grow through the extracellular matrix and apply the sRNAs labeled with Texas red, and we can see here a pollen tube with a lot of sRNAs inside of it. Here at this point is the tip of the pollen tube, and also you can see here a close plot dividing the live part of the pollen tube with the old part. So the sRNAs is here, and look, we can see here very close to the tip a large amounts of the sRNAs. So, but how the sRNAs are incorporated to pollen tubes? We can see here this result using plants that Professor Alice Strong from the University of Massachusetts gave to Luis, and then Luis gave me them, and then asked her to I can use them. So, but they produce these plants. They have a construct that is with a very specific promoter for pollen expression with here with a membrane marker and GFP. So these pollen tubes you can see here growing through the style, and you can see this kind of holes here, and this is the sRNAs labeled with Texas red, and when you merge images, you can see the sRNAs inside this compartment. And when we test what kind of compartment is this, using another transgenic plant expressing a vesicle marker, you can see here how the vesicles are volatilizing with the sRNAs. It means that these vesicles are transporting the sRNAs inside the pollen tube. So, but how this sRNAs is getting inside the pollen tube, we use endocytosis inhibitor, that which is using when you have to prove this endocytosis is mediated by the Catherine pathway, using this T-phosphine. Sorry about that. So, and this is inhibiting the endocytosis, but what you can see here, now using the transgenic plant expressing the membrane marker, when you don't have the inhibitor, the sRNAs are here and also are inside the pollen tube. You can see there in this orthogonal slide, are inside the pollen tube, but when you add the endocytosis inhibitor, the sRNAs still outside the pollen tube. It means that the sRNAs are getting the pollen tube cytoplasm through the clathrin-mediated endocytosis. Also, we have very nice friends, and Bruce McClure at the University of Missouri in USA, he gave me some transgenic plants expressing a vacuum marker. And then when we evaluate how the sRNAs is after internalization in the pollen tube, what is the final destiny. So we have here This pollen tubes moving after 15 minutes to add the sRNAs. We can see here the vacuum. This is the normal shape of the vacuums and pollen tubes, and the sRNAs looks in the same way, same pattern, but this is inside of the vacuum. But 30 minutes later, you can see, you are not able to see a very nice vacuum here, and the sRNAs are dispersed through the whole cytoplasm. Then it means that after this time, the sRNAs are in the cytoplasm, and then we test if this sRNAs is going to degrade the RNA, and we evaluate how the sRNAs integrity after 60 minutes, and we found that the sRNAs are degrading the sRNAs when they are on the cytoplasm. So finally, we have this model. This is a very simple model one, where the sRNAs are getting inside the pollen tube through endocytosis, and then they are going to the vacuum, and this vacuum, after a few minutes, are going to disrupt, and the sRNAs are going to be in the cytoplasm, and they are going to degrade the RNA. So I think that we have a more simple biological model to evaluate how the vacuum is disrupted. I mean, I'm thinking about the morning talk this morning, how other cytoplasmatic changes are implied in triggering some pathway to disrupt the vacuum integrity. So we can test now a lot of things using this approach. And just the last one too. I've noticed a lot of people that have been working with me give me material and give me some very nice ideas to perform it. Thank you very much.
So, just one question. How the RNAs goes into the membrane of the pollen tube? I mean, it has some kind of recognition, or
I think that there is a receptor. Even more in Apple, there is a report where a receptor that is transporting sRNAs inside the pollen tube is working. But this is an Apple. I don't know if this is here also, I mean the same one, but I almost sure that there is a receptor. This is another question that we have to ask.
Yeah. Thank you. It was very nice, and I'm also very curious about the pathway. Like you have you said that this is introduced by endocytosis into the cytoplasm, so you will get that into the vesicles, the endocytic vesicles, and then how it gets incorporated into the vacuum. Is it a fast process? It takes some time. How is it regulated?
But I think that this is a fast process, but I don't know how yet.
Okay.
But I'm almost sure that these bicycles are fusion into the vacuum.
Right. So I can imagine that there is like a specific place at the apical region where most of the endocytosis is taking place. And as we know, the vacuum is very active and it's moving all this time. I was just curious about if there is like a specific places where you can get a fusion of this vesicles with the vacuum.
Yeah. So this is a very, very nice question
That
because
would be very nice to
I have to do this in real time, and I'm thinking in Tatiana, the woman that is working in the Faculty of Sciences, to do this in real time and measure a lot of things. And I think that we will be able to see all the traffic after bicycles, and then how they fuse to the vacuum, and then how the vacuum is
Thank you, Felipe. So our next speaker is Matt Jones from Thermo Fisher Scientific. So I hope just I know we're out of time, but this is a very interesting talk about technical things. Thank you very much.
Thank you, and I'll try to go a little bit quick on this. I'm sure everyone's a little bit ready for lunch. So my name is Matt Jones. I am a technical specialist here at Thermo Fisher, focusing mostly on electron microscopy. And what I'd like to really talk about today is kind of how a lot of this has changed over the last, I've been doing this for about 16 years now, and really a lot has changed over these last few years. So if we think about just the world of plant research and all of the different businesses that have come out of this, no matter what we're talking about, whether it's research in food, fuel, drugs, textiles, at the center of all of this, wrong direction, we are finding that electron microscopes are becoming a very important part of all of this research. And the reason for this is if we look at the overall length scales of different techniques that we like to use in the lab, electron microscopy is very, I think it has a lot of breadth to it because it is able to combine something where we're not only working at very high resolution, but we're able to use this technique to span a lot of length scales. And within this, I'm going to talk mostly about starting with some scanning electron microscopes, and then we're going to talk a little bit about how these are used to actually do a little bit of sample preparation to go into the transmission electron microscope, and I'll be finishing up with a really nice example of some recent research in this area. So one of the kind of drawbacks of any sort of working with just kind of some of the more typical methods of just SEM is if we're talking about so if we're talking about, we'll use a plant pathogen, for example, an instance of wilt here. A lot of times we aren't always able, from our naked eye, able to see what is causing a pathogen like this. If we use light microscopy, we can then oftentimes start to probe this a little deeper, and we can start to see more of what's actually happening inside of our cells. But to go even further with this is where now I think light microscopy and a lot of electron microscopy, they're not necessarily different techniques, but I think something that can really be used together in conjunction to start answering a lot of really important questions. And so we can use these to now see inside some of these cells. But there's an inherent limitation of what we can actually achieve with just a regular scanning electron microscope. So if we are looking at a bacteria, for example, and someone says, what's the average size of all these bacteria? You're limited in what you can do because there's so much overlap of all these structures. Or if we are looking at, say, something like a fungus, and it's actually going through some of these cells. If we're looking top down, then we're kind of missing what's happening at the cell wall level. Same thing within the TEM. We are working with something that is just a 70 nanometer, thereabout section going through a cell. So it's not really the whole cell, it's just a small snapshot. And so if we're looking at the overall structures that we see, it kind of starts to ask the question of, is this really the shape of all these organelles here? Are something like mitochondria really a pill shape, as it looks in this image? Or is it maybe something that's like what a lot of us are seeing with light microscopy, where these shapes might be a little more dynamic? And so we then can consider a technique that's starting to grow a little bit more, known as volume electron microscopy. And simply put, this is where we are able to look at our cells and tissues at nanometer resolution, but now we can extend this into several microns in that Z direction. And with this, we have plenty of resolution to identify all of the different organelles that exist within these cells, but we can take it one step further. And I think the real power of this is going through and being able to put our cells together piece by piece, organelle by organelle, to really start to see why all of these structures are related to one another. Or for example, if you have something like a ciliate structure, why do all those mitochondria sit there? Is it maybe because it's a little bit power hungry? Now, when we talk about volume techniques, you've probably seen a lot of different names in research, and pretty much they're all the same thing in the sense that the idea here. Now, this first started with something known as array tomography, and I think this is kind of the easiest and quickest way to get into doing our 3D imaging, where you take an external ultramicrotome, like a lot of us have worked with, but instead of putting all these sections onto a TEM grid, we can put it onto a conductive substrate, like silicon. We can then use our SEM to go ahead and image each of those sections, the same area on each of those sections, and then we can use that to build up our model. Now, serial block face, as there have been a few people that have talked about this this week, this is where we then took that ultramicrotome and we shrank it down so it's small enough to actually fit in the chamber of our SEM. And then we can use the SEM to just look at the exposed block face surface to start generating all of these images. But, has anyone here done sectioning before? It's a little bit tricky, and as I'm sure most of you have found, there's a limit to how thin you can go with our sections before things start going a little bit wrong. So to kind of bypass that Z resolution limit, we actually are going to talk about a little bit of a different microscope, something called a FibSem, where we are able to do two different techniques to build this up. One is called slice and view, where we use this microscope to actually dig a hole into our sample, and we actually are able to look at that cross-sectional approach to generate all these images, or something new called spin mill, where we're actually using the microscope to polish off the very top of our sample, so that again we can start to build up these 3D data sets. Now, starting with serial block face, so this has actually enabled a lot of really, really exciting research in the last few years. So one of my favorite examples, a group that recently was looking at the distribution of plasmodesmata in a sample that was had some sort of a viral infection. And we can see that one of the great things about this is, I had mentioned earlier the kind of extreme length scales that this is capable of. So instead of just looking at single snapshots here, we can take this whole volume and now start to build it up and actually have kind of an idea about the distribution of these different PD structures. But, as I mentioned, there is a limit. So if we start looking at some of these orthogonal projections here, we can definitely visualize all of these PD structures. But if you want to do anything beyond just counting them, you maybe want to see a little bit more of that dynamics, where it's somewhat limited in the resolution of what we have to work with. So this is where that other technique, working with what's called a FibSem, comes in, and this is able to give us actually the same resolution in X, Y, and Z, so that we are able to slice these samples any way we want once it's collected, and we still have very, very high resolution about what's happening in these samples. So this is actually requiring